And first live session we are going to doing, and
Stop faking your accent.
we will study in this live session, that is sexual reproduction and flowering plants, the remaining part, and you can call it the end game. Okay, because it is the last one, means it's the finishing part. So so I'm starting to study the remaining part. But before that, your macho boy will revise you the entire that you studied yesterday. Okay, so let's start.
আর আই প্রিফার দোজ যে ভিডিওটা প্রথম থেকে পরে দেখবে লাইভ খুলেই এরকম
Flower, a fascinating organelle in this poem. Flower is a modified shoot which allows favorable adaptation for reproduction through spatial method. Flower consists of calyx, corolla, androecium, and gynoecium. हेलो, कैसे
चिन्मय
हो?
सर
Emmanuel R. Hi bro. Yuna Mega. Hello. How are you all, my dear
চিন্ময় স্যার
peoples?
চিন্ময় স্যারের তোকে গ্রুপে পড়েছিস তো তাই জন্য না তোর ভালো লাগে না।
Hello, so let's start. Flower, a fascinating organ of angiosperm. Flower is a modified shoot which shows favorable adaptation for reproduction through a special method. Flower consists of calyx, corolla, and rhizium and gynoecium. Flowers are morphological and embryological structure and are the sites of sexual reproduction. Flower consists of calyx, corolla, and rhizium and gynoecium. What are monocarpic plants? Monocarp the plants in which the flowering and fruiting takes place only once in their whole lifespan are called monocarpic plants. Example: annual and biennial. Annual, annual and biennial. Annual means the plant which, after producing one flowering in their lifespan and die within 1 year, and biennial, after producing once in the lifetime of flowering and fruiting, they die within 2 years. Polycarpic plants. এটাও নেই এনসিইআরটিতে, এটাও নেই এনসিইআরটিতে polycarpic plants. তাহলে the plants in which flowering and fruiting takes place many times in their entire lifespan are known as polycarpic plants. Okay, entire lifespan में बहुत बार ये लोग पैदा करते हैं flowering or fruiting को. Okay.
Guys, I'm sorry for your ears. Protected at any cost.
So, bamboo, palm, banana, century plant, bamboo, palm, banana, and century plant are perennial plants, but they are ওই আর তুই নিয়ে নেন. Bamboo palms, banana, century plant are perennial plants, but they are examples of monocarpic plants. Pre-fertilization structures and events. Okay. So androecium consists of wall of stamens. Gynoecium, female reproductive structure, consists of ovary, style and stigma. Stamen, filament, long, thin and slender stalk-like structure. And anther, swollen spore-bearing structure. Anther, diapicus, bilobed, tetragonal structure. Whole microsporia located at corners to each lobe. Anther, bilobe, pollen sac, pollen grain, tetragonal structure, typical stamen, three-dimensional cut section of an anther. Structure of microsporidium. The microsporidium develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. They extend longitudinally all through the length of an anther and are packed in pollen grains. Microspine is surrounded by four wall layers: epidermis, middle layer, epithelium, and tapetum. Cells of the tapetum possess 10 cytoplasm and more than one nucleus. Storage of pollen grains -196 degree. Viability 30 minutes in rice and wheat per month in some members of Rosaceae and Leguminaceae. Pollen grain walls of pollen grain exine outer layer thick, hard and rigid, made up of sporophyllin in most cysteine organic material known. Intine inner layer thin and continuous layer made up of cellulose and pectin. Dehiscence at 2-cell stage in over 60% and angiosperm at 3-cell stage in the remaining species. Genitive cells smaller and float in the cytoplasm of vegetative cells, spindle shape with dense cytoplasm and a nucleus. Vegetative cell bigger and has abundant food, preserve large irregularly shaped nucleus. Size of the pollen grain is 25 to 50 micrometer in diameter. Shape is generally spherical. Okay, can be used as pollen banks in crop production management. Okay, largely illumination vegetative cell stage. Okay. Then we have advantages, rich in nutrients, used as food supplement, increase the performance of athletes and racehorses. Disadvantage causes allergy and bronchial applications that may lead to chronic respiratory disorder. Parthenium or carrot grass came into India as a contaminant with imported wheat causes pollen allergy. The pistil megasporidium and the ovule endosac. The free unit of gynoecium is called pistil or carpel. Okay, this we will read after. Pollination is a process, transfer and deposition of pollen grains from anther to the stigmatic surface of the flower is called pollination. It is essential to prevent sexual reproduction of seed-bearing plants. There are two main types of pollination: self-pollination and cross-pollination. Cross-pollination, xenogamy or allogamy. Flowers from different plants of the same species, pollen grains are transferred to the stigma of the flower or another plant. It occurs in unisexual flowers. Xenogamy, flowers of different plants of the same species. Flower pollen grains are transferred to stigma of the same flower of the another plant. It occurs in unisexual flowers. Self-pollination, pollen grains are attached on the same flower or another flower borne of the same plant. It occurs in bisexual flowers. Autogamy, same flower, pollen grains are transferred to the stigma of the same flower. Latonogamy, different flowers of the same plant. Flowers of different species of the same plant. Flowers of different plants of the same plant. And self-pollination, autogamy, pollen grains transferred from the same flower within the same flower. Okay, dichonogamy, cross-pollination, different flowers of the same plant, pollen grains transferred to anther, another flower born on the same plant. Pollen grain transferred to another flower born on the same plant. Pollen grains transferred from different flowers on the same plant. Flowers from different plant of the same species. Flowers from the within the one flower transferred to the stigma of the same flower. Flower of the same different flower of the same plant. Self-pollination. It is the process of transfer of pollen grains from the anther to stigma of either the same flower or another flower born on the same plant. Cleistogamy. Plants remain closed to ensure complete self-pollination. They are called shy flowers. Okay. So the flowers are small, colorless and do not secrete nectar. Commelina, Venadensis oxalis viola, Pomodina oxalis viola. Casmogamus flowers are normal but solo, okay, like they only want to talk with themselves. Shy flower, they are busy in mating with itself, okay, by giving high five. They are bud pollination, teenage marriage. Before the actual flowers come, the pollination occurs within the bud. Before the actual flower comes, the pollination occurs within the bud. To ensure self-pollination, wheat, rice, pea. Homogamy, maturation of anther and stigma of a flower at the same time is called homogamy. As a rule, homogamy is essential for self-pollination. As a rule, homogamy is essential for self-pollination. So, perfect timing, perfect clock, mature at the same time. Advantages of self-pollination. Chances of pollination are more. Self-pollination maintains purity of the race and avoid mixing. It need not produce a large number of pollen grains. Flowers need not possess device such as large and showy petals, presence of nectar and scent, etc. to attract pollinator. Disadvantage: progeny continuously gets weaker after every generation, less chances of the production of new species and variety. As adaptations in flowers for pollination. Pollination by wind is more common amongst abiotic pollinations. Both wind and water pollinated flowers are not colorful and they do not produce nectar. Both wind and water pollinated flowers are not colorful and they do not produce nectar. Pollination by wind is more common amongst abiotic pollinations. Types of pollination on the basis of pollinating agent. Wind, abiotic agents, wind and water, biotic agents, insect, birds, bats, snails and ants. Micromephily ants, macrophage snail. Okay. Wind, water, anomophily, hydrophily. Insects are known as entomophily, orthiophily. Wind pollination, pollen grains light, non-sticky, winged, easily transported. Pollen light, non-sticky, winged, easily transported. Stamen, well exposed stamen in wind pollination so that pollen grains are easily dispersed. And stigma large and feathery. Flower, one ovule arranged as inflorescence, example corn cob. One ovule arranged as inflorescence, example corn cob. Pollen grains are in light, non-sticky, winged, easily transported. Stamen well exposed so that pollen grains are easily dispersed. Stigma large and feathery. Water pollination, hydrophily, bryophyte, pteridophyte, algae. Pollen grain protected by mucigens covering, example freshwater, vallisneria and hydrilla. Sea grasses. Sea grasses, Zoostera, produce pollen grains in large number, do not produce nectar. Female flower remains submerged. Pollen grain long ribbon-like, released inside water, carried by water currents to reach female flower. For vallisneria only, the female reach the water surface by long coiled stalk. Male and pollen grain released onto the water surface, carried by water current. Some events reach the stigma. সো ভ্যালিসনেরিয়াতে কী হচ্ছে, ফিমেল ফ্লাওয়ার ওয়াটার সারফেস থেকে উঠিয়ে চলে আসছে যাতে পলিনেশন হতে পারে পোলেন গ্রেনস। কিন্তু জুস্টেরাতে ফিমেল ফ্লাওয়ার সাবমার্জ আন্ডারই থাকে, লং রিবন লাইক আর রিলিজড ইনসাইড ওয়াটারই হয়। ঠিক আছে, cross certain rivers to pollinator, nectar and edible pollen grains as food, provide safe place for laying eggs, example Amorphalus and Yucca. Example, Amorphalus and Yucca. Nectar and edible. Certain flowers, large insect pollination, large colorful, fragrant, rich in nectar. Pollen grains are sticky and stigma is sticky. Amorphalus and Yucca. Although geitonogamy is functionally cross-pollination, geitonogamy is functionally cross-pollination involving a pollinating agent. Genetically, it is similar to autogamy since the pollen grains comes from the same plant of the different flower, another flower born on the same plant. So geitonogamy cross pollination, but here the pollen grain is coming from the same plant. Pollination by water is quite rare in flowering plants and limited to about 30 genera, mostly monocotyledons. Bud pollination. Bud pollination, flowers occurs inside the bud even before the flowers open to for teenage marriage. Self-pollination, plant tindered, swine, knife, monoacid. Cleistogamy, closed for business. Okay, cleistogamy. Even closed ensure to complete self-pollination, small, colorless and do not emit nectar. Camelina, oxalis and viola. Bud pollinator, wheat, rice and pea, etc. Bud pollination. Casnogamas. Homogamous, sync your flock. Outbreeding devices. Outbreeding devices are mechanisms which are followed and adapted by the plant in order to prevent self-pollination, continuous self-pollination resulting in breeding depression. Another anther and stigma placed at different position. Outbreeding devices, self-incompatibility, prevent self-pollination from fertilizing ovules by inhibiting pollen germination or pollen tube growth in the pistil. Pollen grain release and stigma receptivity are not synchronized. Pollen is released before stigma becomes receptive, and stigma becomes receptive much before the release of pollen. Production of unisexual flower. Monocotyledonous plant, both male and female flowers are present on the same plant. Example, castor and maize. Dioecious plant, male and female flowers are present on different plants. Example, papaya. This condition prevents both autogamy and geitonogamy. Male and female flowers are present on different plants. Example, papaya. Both male and female, example castor, maize. Both male and female present on different plant. Example, papaya. This condition prevents autogamy and geitonogamy. Autogamy. Same flower, geitonogamy. Same plant but different flower. And the synogamy, flower from different plants of same species. Self-pollination, transfer to the stigma of the same flower or another flower born on the same plant. Zygomys. Autogamy is pollen grains are transferred from the stigma of the same flower. Geitonogamy, self-pollination is of two types. Autogamy is not either the same flower or born on the same plant. One and one, two. One and one, two. Synogamy of different plant of the same species. Pollen pistil interaction. All events from deposition of pollen on stigma till the pollen tube enters the ovule. These are deformation of compatible pollen, germination of pollen grains, development of male gametophyte. Artificial hybridization, it is one of the major approaches of crop improvement program. The desired pollen grains are used for pollination and the stigma is protected from contamination. And this is achieved by two techniques: emasculation, removal of anther from the flower bud before implantation, bagging, flower covered by a bag made up of butter to prevent contamination of stigma with unwanted pollen. Bag flower attains receptivity, matured pollen grains and resting on the stigma, and the flowers are rebagged and fruits are allowed to develop. Double fertilization is the process of fusion of one male gamete with egg along with the union of second male gamete with the two polar nuclei of the secondary nucleus. Double fertilization is a unique event to flowering plants, NGS form only. N-gamete, 1 Excel and triple fusion. Second male gamete, 2 polar nuclei, pain 3 and C-gamete is known as 1 male gamete if the egg cell and triple fusion equals to double fertilization. The central cell after triple fusion becomes the primary endosperm cell and develops into the heterosperm while the zygote develops into an embryo. Now we will read about post-fertilization structure and events. Then at the last we will study testing. Before that, 3 minutes of just break. Okay. So you now I'm sitting here for So let's start. Post fertilization structures and events. Events of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed as post-fertilization events. Events of endosperm and embryo development, maturation of ovule into seed and ovary into fruit. Events of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed as post-fertilization moments. So seed, ovule develops into seed, ovary develops into fruit, and endosperm developed. Endosperm. Development of endosperm takes place before the embryo development. Development of endosperm takes place before the embryo development. Okay. Primary endosperm cell divides repeatedly to form a triploid endosperm. Primary endosperm cell divides repeatedly to form triploid endosperm. Cells are filled with reserved food materials and are used for the nutrition of the developing embryo. डेवलपिंग एम्ब्रो के लिए न्यूट्रिशन यूज़ करता है. और जो है पेन अंडरगो सक्सेसिव न्यूक्लियर डिवीजन to give rise to free nuclei. This is called free nuclear endosperm. Subsequently, cell wall formation takes place and becomes cellular endosperm. Subsequently, cell wall formation takes place and becomes cellular endosperm. Development of endosperm takes place before the embryo development. Primary endosperm cell divides repeatedly to form a triploid endosperm. Cells are filled with reserved food materials and are used for nutrition for the developing embryo. Okay, for developing embryo. What is embryo now? Endosperm complete. Development of endosperm takes place before the embryo development. Primary endosperm cell divides repeatedly to form a triploid endosperm. Cells are filled with reserved food material and are used for nutrition of the developing embryo. Pain undergoes successive nuclear division to give rise to free nuclei. This is called free nuclear endosperm. Subsequent cell wall formation takes place and becomes cellular endosperm. Embryo. Zygote formed and placed at micropillar end of the embryo sac. Zygote formed and placed at the micropillar end of the embryo sac. Zygote called primary endosperm cell. Zygote formed and placed at the micropillar end of the embryo sac. Zygote called primary endosperm cell. Zygote starts its development only after some amount of endosperm is formed. This is an adaptation to provide assured nutrition of the developing embryo. So zygote फॉर्म होकर माइक्रोपिलर एंड में एम्ब्रो सैक में चला गया. zygote को बोला जाता है प्राइमरी एंडोस्पर्म. zygote बुलाता है प्राइमरी एंडोस्पर्म सेल को. zygote तब तक डेवलप नहीं होता जब तक कि एंडोस्पर्म डेवलप नहीं हो जाता, क्योंकि वो उसको न्यूट्रिशन देगा. मोनोकॉट एम्ब्रो वर्सेस डाइकॉट एम्ब्रो. Consist of embryonal axis and two cotyledons. Consist of embryonal axis and two cotyledons. Embryonal axis above the cotyledon is the epicotyle. Embryonal axis above the cotyledon is epicotyle. Terminal part of the epicotyle embryonal axis below the cotyledon is the hypocotyle. Embryonal axis terminal part of the hypocotyle is radical orbit. The root tube is covered by root cap. radical dicot structure. Immune. Cotyledons. Embryonal axis above the cotyledon is epicotyle. Embryonal axis above the cotyledon is epicotyle. Terminal part of the epicotyle is plumule, and below embryonal axis below the cotyledon is hypocotyle, radical and root tip. So consists of embryonal axis and two cotyledons. Embryonal axis above the cotyledon, cotyledons above the cotyledon is epicotyle, and below the cotyledon is hypocotyle and radical and root cap, and upper above the epicotyle is plumule. Okay, so plumule ये structure हो गया. Monocot embryo possesses only one cotyledon. In grass family, the cotyledon is called scutellum. Possesses only one cotyledon. Okay, possesses only one cotyledon, and in grass family, the cotyledon is called scutellum. In grass family, the cotyledon is called scutellum. Yes, Rao Sahab. Sexual reproduction flowering plants. Monocot embryo possesses only one cotyledon. In grass family, the Okay, let's draw the structure. Scutellum. Coleoptile. Shoot apex. Epiblast. Radical. Root cap. Polerohija. Plumule. Below the plumule, epicotyle. cotyledon, terminal terminal part of the epithelium, and below the cotyledon is hypophyte, and there is a radicle, and also there is a root cap. This structure is dicot structure. Monocot structure. Scutellum, coleoptile, cleoptile, shoot apex, epiblast, radical, root cap, coleoptile. That's it. Scutellum, coleoptile, shoot apex, epiblast, radical, root cap, coleoptile. The coconut water is free endonuclear sperm, and the white kernel is free endonuclear sperm and cellular endosperm. Embryo development takes place in following stages: zygote, proembryo, globular embryo, heart-shaped embryo, matured embryo. Zygote, proembryo, globular. Heart, mature. Zygote, proembryo, globular embryo, heart-shaped embryo, and mature embryo. Post-fertilization changes. Shoya Tom sir. Hi. हाय शौर्या तम एसआर कहाँ से हो आप? Where are you from, Shreya Thamsar? Okay, Chandigarh. Okay, very good. Thank you, Shreya Thamsar. Best of luck to you also. The coconut water is free nuclear endosperm and white kernel is cellular endosperm. Embryo development takes place in the following stages: zygote, proembryo to globular embryo, globular embryo to heart shape to mature embryo. Post fertilization changes. After fertilization, the sepals, petals, stamen, style, and stigma with either fall off. The calyx may persist, example tomato brinjal, and even show growth physalis. Post fertilization changes after fertilization. Oh, you are also a YouTuber. That's good. What is your channel name? Is this here the channel you use? Post fertilization changes. The calyx may persist, tomato brinjal, and even show growth physalis. Pre fertilization structure and post fertilization structure difference. Now we will study. Pre fertilization ovary, post fertilization structure fruit. Ovary wall, pre fertilization pericarp. Ovule develops into seed. Funicle stalk of the seed. Ileum. Nucellus perisperm. Outer integument, testa seed coat. Inner integument, tegment seed coat. Micropyle, fertilized egg, endosperm, synergies and antibodial cells degenerate, fertilized secondary nucleus. So ovary, fruit, ovary wall, pericarp, ovule, seed, funicle, stalk, ileum, nucellus, perisperm, outer integument, inner integument, outer integument, tegment, testa, inner integument, tegment, microbial, fertilized egg, endosperm, synergies and antibodial degenerate, fertilized and egg and secondary nucleus. Pre-fertilization structures and events. Pre-fertilization structures and post-fertilization events. Difference is ovary, fruit, ovary wall, pericarp, ovule, seed, funicle, helium, nucleus, perisperm, outer integument, testa, inner integument, tegment, microbial, fertilized egg into endosperm, synergids and antipodal cells degenerate, fertilized secondary nucleus. So ovary, fruit, ovary wall, pericle, funicle, stalk of the seed, helium, nucellus, perisperm, outer integument, testa, inner integument, tegment, micropyle, fertilized egg, endosperm, synergid and antipodal cells degenerate, fertilized secondary nucleus. Now we will study seed. Seed is the final product of sexual reproduction. Seed is the final product of the sexual reproduction. Seed consists of seed coat, cotyledons and an embryo axis. In a made seed, scutellum is considered as cotyledon because it absorbs food material and supplies them into embryo. In a made seed, scutellum is considered as cotyledon. In maize seed is example of scutellum is considered as cotyledon because it absorbs food material and supplies to the embryo. In maize seed, scutellum is considered as cotyledon because it absorbs food material and supplies them to embryo. In legumes, the cotyledon of the embryo are simple structure, generally thick and swollen due to storage of food reserves. In legumes, the cotyledons of the embryo are simple structure, generally thick and swollen due to storage of food reserves. In maize, the scutellum is considered as cotyledon because it absorbs food materials and supplies them to embryo. Seed, non-albuminous or ex-albuminous seed, albuminous seed. They have no residual non-albuminous seed. They have no residual endosperm as it is completely consumed during embryo development. Example: pea, groundnut, sunflower, bean. Pea, groundnut, sunflower, bean. Pea, groundnut, sunflower, bean. Non-albuminous seeds. Pea, groundnut, sunflower, beans. Albuminous seed. They retain a part of endosperm and it is not completely used up during embryo development.
দিয়ে আসবে, হ্যাঁ আসছে, হ্যাঁ ওটা এটা কি ফটো করে রেখেছে, কি সুন্দর, হ্যাঁ এটা কী করে করে সবসময় উঠছে। তোমার তো পরীক্ষা এলো, আর তো 1 মাস 30 দিন বাকি, কতটা তৈরি হলো
হয়েছে?
হ্যাঁ ভালো হয়েছে সব। আবার পরীক্ষা কবে আছে? পরীক্ষার রেজাল্ট বেরিয়েছে?
হ্যাঁ,
কত?
ওই তোমার 500 এখন।
বেশি আর উঠছে না, মানে এখন কি ফুল সিলেবাসে হচ্ছে? দেখি তাহলে তোমার 500 550 আশপাশেই তো উঠছে বরাবর। তো ওটাই 600 হবে। হ্যাঁ কিরম আশা করো, মানে
কি আমি দেখি,
হবে তো না?
পরীক্ষার পরই বলবো।
আচ্ছা ঠিক আছে। আর এটা পারোটা একটু খুলে দাও নাকি?
না এখন রোদ।
আচ্ছা ঠিক আছে।
Okay, only 31 minutes left. And albuminous seed, non-albuminous seed, they return a part of endosperm and it is not completely used during embryo development. Example: wheat, maize, barley, castor, coconut. Wheat, maize, barley, coconut, castor. Classification of fruits: fleshy, guava, mango, mango. Fleshy fruits: guava, mango. Dry: groundnut and mustard. False fruit: this fruit develops from the floral parts. Apple, strawberry, cashew develops from thalamus. These fruits develop from other floral parts. Example: apple, strawberry, cashew develop from thalamus. True fruits: this fruit develop only from ovary. True fruits develop only from ovary. So, fleshy fruits. Remnants of nucellus are also persistent: guava, orange, mango. Guava, orange, mango. Dry fruit: groundnut, mustard. False fruit: apple, strawberry, cashew develops from thalamus. Apple, strawberry, cashew. Apple, strawberry, cashew. True fruits: this fruit develop only from ovary. True fruits: this fruit develop only from ovary. True fruits. Classification of fruits: this fruit develops from other fleshy guava, orange, mango, groundnut and mustard. Apple, strawberry, cashews. True fruits develops from ovary. That's it. Remnants of nucellus are also present in some seeds is called perisperm. Black pepper, beet. Remnants of nucellus. Black pepper, beet. अरे आजा ना
সেলফি স্টিকটা দিয়ে এরকম ভাবে ধরে চাচা চাচা করে তোলা যায়। টাইমার টাইমার দিয়ে এরকম করতে হবে।
টাইমার দিয়ে
কেন এটা টিপে তোলা যায় না?
না রে
কেন
তোলা যায় না।
তাহলে ওই একটা কি খোলা পড়ানোর সিস্টেমটা কি?
ওটা এমনি দিয়েছে শো।
আমার কাজেই লাগে না। আমার ভালো সেলফি স্টিক নেই। ভাবছি তাহলে এটা কিনবো।
কেন
পরপর ক্লিকসগুলো কাশ্মীর দিয়ে না চাইনিজ সাথে শুরু করতে হবে। তো রাখতে তো হবে একটা
তাহলে ওটা কাজ করে, আমি কাজ যখন করতে দেবো না তোকে ওটা।
কেন?
ব্যাটারির ওটা শেষ হয়ে গেলে আমার ব্যাটারি আর কিনতে পাওয়া যায় না, অনেক দাম ওটা, 86 টাকা একটা ছোট ব্যাটারির দাম।
কি যায় আছে?
কি যায় আসে, তুই এখন ইচ্ছা করে শেষ করবি ওটা?
ইচ্ছা করে না।
ওটা শুধু আমি ইউজ করব
এরম
ওই
কেন? তোর
ফিচারটা।
মনোটনই কেন?
ওটা আমি দেবো না ফিচারটা ইউজ করতে তোকে, আমি
জানলা
সেলফি স্টিক পুরো কেড়ে নেবো না হলে যদি ওটা ইউজ করিস।
কাশ্মীরে গিয়ে ওটা use করবো।
ঠিক আছে,
ওইটাই আমি বলেছি, মানে এখন নিজের থোবড়া তুলবা না selfie
না
stick।
জেনে গেলে বলে দিলে নিজেই করতে শুরু করে দিবি,
ও,
তোকে
এর
তো
হলো
চিনি।
পরিচিত। আমি মিথ্যের আশ্রয় নিচ্ছিস।
কারণ তোর ওপর দিয়ে রেখেছি না।
ঠিক আছে, কাশ্মীরে গিয়ে জানিস। আমার ঘরটাকে সবাই বলছে হোস্টেল, তোর ওই দেয়ালের জন্য। হ্যাঁ, তোকে বলো মাঝে দেয়াল ফেয়াল দেখে দুজন বলছে হোস্টেল। আগে আগে বলছি।
হুম। Okay, so in a maize seed, scutellum is considered as cotyledon because it absorbs food material and supplies them to the embryo. In legumes, the cotyledons of the embryo are simple structure, generally thick and swollen off due to food reserves. Okay. In legumes, the cotyledons of the embryo are simple structure, generally thick and swollen due to soil. In maize seeds, scutellum is considered as cotyledon because it absorbs food. So pericarp remnants of nuclei are also persistent in some seed is called perisperm, black pepper beet. The wall of the tree develops into wall of fruit and called pericarp. Integuments of ovules, hard and tough protective seed coat. The microbial remains a small pore in the seed coat. They facilitate the entry of oxygen and water into the seed during germination. Advantages of seed to angiosperms. Since reproductive phase such as pollinates and fertilization are independent of water, seed and formation is more dependable. Since reproductive process such as pollination and fertilization are independent of water, since reproductive process advantages of seeds to advantage. Since reproductive process such as pollination, fertilization are independent of water, seed formation is more dependable. Seeds have better adaptive strategies for dispersal to new habitats and help the species own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combination leading to variation. Being products of sexual reproduction, they generate new genetic combinations leading to variation. The oldest viable seed of a lupine, Lupinus arcticus, excavated from Arctic tundra, the seed germinated and flowered after an estimated record of 10,000 years of dormancy. Lupine lupinus are excavated from Arctic tundra. The seed germinated and record of 10,000 years of dormancy. A recent record 2,000-year-old viable seed is date palm Phoenix dactyphora discovered in the archaeological excavation at King Herod's Palace near the Dead Sea. Parasite species such as Orbonia and Striga. King Herod's Palace. King Herod's Palace. King Herod's Palace. Near Dead Sea. Discovered Phoenix daclylifera. Phoenix daclylifera, which discovered and successfully grown from the ancient seeds found near the 1960 archaeology discovered 200-year-old date palm seed at Masada, an ancient fortress near the Dead Sea. The seeds were stored for centuries and these seeds were stored for centuries and later kept in research facility. In 2005, Dr. Sarah Saloon team successfully germinated one of these seeds, naming the resulting tree Methuselah after biblical figure known for his longevity. Further display led to the growth of additional date palm from ancient seed, providing insight into ancient agriculture and plant resilience. The oldest viable is lupine. Parthenocarpic fruits. The development of fruit is an unfertilized flower. The development of fruit is an unfertilized flower without fertilization, resulting in a seedless fruit. It may occur naturally as in certain varieties of pineapple, grapes, apple, beer and banana. Unfertilized flower is known as fruit, means without fertilization. Parthenocarpy may be induced by application of plant growth hormone as in tomato. Thalamus seed. Endocarp, mesocarp. Okay. So the development of fruit in an unfertilized flower without fertilization resulting in a seedless fruit. It may occur naturally as in certain varieties of pineapple, grapes, apple, beer, pear and banana. Parthenocarpy may be induced by application of plant growth hormones as in tomato. Last is apomixis. It is a form of asexual reproduction, mimics sexual reproduction. Apomixis hybrid common in astrocracy and gases. Method apomixis hybrid plants developed by this method maintains the genetic identity. Diploid egg cell formed without reduction division and develops into embryo without fertilization. Diploid egg cell formed without reduction division and develops without fertilization. Embryo develops without fertilization is known as apomixis. Astrocracy and gases are the common example. Astrocracy and gases are the common example. Okay. Now polyembryony occurrence of ovule having more than one embryo in a seed is referred to as polyembryony. Some of the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac and develop into embryos. In such species, each ovule contains many embryos. Example: mango and citrus. Mango and citrus have many embryos. Mango and citrus have occurrence of ovule having more than one embryo in a seed is referred to as polyembryony. Some of the nuclear cells surrounding the sac are protrude into embryo sac and develop into embryos. So many embryo, okay, one embryo in a seed. Problems associated with hybrid varieties of food and variety. Hybrid seeds produced every year. If the seed collected from hybrids are sown, the plants in the progeny segregate and do not maintain hybrid characters. Production of hybrid seeds is costly and hence the cost of hybrid seeds become too expensive for the farmer. If these hybrid seeds are made into apomix, there are no segregation of character in the hybrid progeny, then the farmer can keep on using the hybrid seeds to raise new crop year after year and he does not have to buy hybrid seeds every year. If these hybrids are made into apomix are correct, there is no segregation of characters in the hybrid progeny, then the farmers can keep using the hybrid seeds to raise new crop year after year and he does not have to buy hybrid seeds every year. Okay, so 90% of the chapter is done except pistil. So thank you. We have learned from post-fertilization structure and events to seed, classification of fruits, apomixis and polyembryony. So in just 5 to 10 minutes I will explain all the structure, all of this, okay, all what we have read. Events of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed as post-fertilization events. Endosperm development of endosperm takes place before the embryo development. Primary endosperm cell divides repeatedly to form a triploid endosperm. Cells are filled with reserved food materials and are used for the nutrition of the developing embryo. Pain undergoes successive nuclear division to give rise to free nuclei. This is called free nuclear endosperm. Subsequently, cell wall formation takes place and becomes cellular endosperm. Embryo zygote formed and appraised that takes place at micropillar end of the embryo sac. Zygote called primary endosperm cell. Zygote starts its developing only after some amount of endosperm form. This is an adaptation of provide assured nutrient to the developing embryo. Monocot embryo versus dicot embryo. Dicot embryo consists of an embryonal axis and two cotyledons. Embryonal axis above the epicotyle. Terminal part of the epicotyle is premiule. The embryonal axis below the cotyledon is the hypocotyle. The embryonal axis terminal part of the hypocotyle is called the radicle or root tip. The root is covered by the root cap. Monocot embryo possessed by only one cotyledon. In grass family, the cotyledon is called scutellum. Scutellum is situated towards one side of the embryonal axis. Radicle and root cap enclosed by sheet called colerohyja. The portion of the embryonal axis above level of attachment called scutellum is called epicotyle. Epicotyle is shoot apex of linear coleoptile. Scutellum, coleoptile, shoot apex, epiblast, radical, root cap, colerohyja. The coconut water is free nuclear endosperm and the white kernel is a cellular endosperm. Embryo development takes place in following stages: zygote, proembryo, globular embryo, heart-shaped embryo, mature embryo. Post-fertilization changes after fertilization, the sepal petal, stamen, style and stigma usually wither and fall off. The calyx may persist, tomato, brinjal.
I'm your teacher, your mentor, your physician का हम सफर दिलजोत सर और इस सफर में खजूर आज हम लोग लेंगे लेक्चर नंबर 2 पे इलेक्ट्रोस्टैटिक कैपेसिटेंस पोटेंशियल ये सारी चीजें हम पढ़ेंगे सो भैया आज कैपेसिटेंस की भी बारी है आज कैपेसिटेंस भी डिस्कस किया जाएगा पर उससे पहले मैं पोटेंशियल पे और डिस्कशन करूंगा लास्ट लेक्चर में अगर आपने चीजें पढ़ी तो आप लोगों को पोटेंशियल निकालना सिखाया था मैंने पॉइंट चार्ज का सिस्टम ऑफ चार्जेस का पोटेंशियल एनर्जी निकालना हमने सीखा था स्केलर क्वांटिटी हम लोगों ने ये सीखा था ये सारी बातों पे हम लोगों ने डिस्कशन किया था पर आज डिस्कशन हम लोग करेंगे पोटेंशियल डी डाइपोल के भी ट्यूटोरियल में मैंने बता दिया था और एक्सेल पे आप लोगों से पूछा था कैसे निकाल सकते हैं डिफरेंट ऑब्जेक्शन उनके लिए पोटेंशियल निकालना सीखेंगे यहाँ पे डिस्टेंस बिटवीन द टू चार्जेस इज 2a एंड ईच चार्ज हैज अ मैग्नीट्यूड q the electric potential due to dipole at a point d distance away from the origin on the equatorial plane.
So, post-fertilization event after fertilization, the sepal petal, stamen, style and stigma usually wither and fall off. The calyx may persist, tomato brinjal even show growth physalis. In a mate seed, scutellum is considered as cotyledon because it absorbs food material and supplies them to embryo. In legumes, the cotyledons of the embryo are simple structure, generally thick and swollen due to storage food and structure. Food reserve. Classification: non-albuminous seed, they have more residual endosperm.
এরাই একটা চামচ আনবি। না কি রে সালা। সব ভিডিও খুলছিস কেন? In milkweed, scutellum is considered because it absorbs food material. In legumes, cotyledons and row are simple structures, thick and swollen due to food reserve. Non-albuminatory, those have no residual endosperm. Pea, groundnut, sunflower, beans. Pea, groundnut, sunflower, beans. Albuminous seed, wheat, maize, barley, castor, coconut. Wheat, maize, barley, castor, coconut. Pea, groundnut, sunflower, bean. Astera and grasses. Mango and citrus polyembryony. More ovules. Okay. तो ये चैप्टर हो गया. अब इसका सिर्फ PYQ बाकी है जो कि अलग सेशन करेंगे उसका. चलिए अभी बाय. दूसरा सेशन में जूलॉजी पढ़ेंगे.